Hello once again. This time I will be doing my third Teach Me Something assignment on the role of histones. And as I am a bit shaky on this subject, hopefully I can try to do my best here at explaining what they are and the function of histones. Let's get started here. What are histones overall? So Histones are going to be the base or the foundation of the chromatin structure as the DNA double helix wraps around them so that they can be highly condensed, as this will allow the DNA to be able to fit inside that nucleus where it's going to be transcribed later to RNA. And secondly, histones are going to be a small group of basic proteins. Concerning this, histones are going to be made like most proteins within a cell. So that means they're going to be made within the cytosol, being synthesized by ribosomes, but they will be imported into the nucleus because that is where they'll be used for the process of packaging DNA, which is where DNA originates. So histones are going to contain high amounts of lysine and arginine. So these amino acids will give the histones a positive charge, which allows them to bind and wrap around the negatively charged DNA double helix. So to get a picture of what histones look like, uh, here is a figure, and as you can see, that DNA double helix, uh, which is the basic structure of DNA, will wrap around these histones as it's going to be getting more compact and it's going to be more packaged. As for the rest of these things that are pointed out in this figure, I will be trying to explain my best here for the rest of my presentation. So for the structure of histones, it is thought that the histones are made up of a family of proteins, and this is divided into 5 main types, which are the H1, H2A, H2B, H3, and H4. Now, only 4 of these molecules will be used to make the octamer, the prefix meaning 8, and the function of this octamer will be what the DNA double helix will wrap around. However, which 4 of the 5 will be the question. Well, in this case, I found in my textbook that the molecules H2A, H2B, H3, and H4 will be the ones consisting of the octamer structure. Now, what about the H1 molecule? Well, according to the book, this is going to be that molecule that is associated with the linker region and will function in binding to the linker DNA and facilitate the packing of nucleosomes into the 30 nanometer chromatin fibers. Additionally, I found in my textbook that a chromatin will have approximately equal numbers of H1, H2A, H2B, H3, and H4, and only half the number of H1 molecules. So I'll provide a figure here in a little bit to explain that. So lastly, these histones that make up the octamer will have tails that protrude from them. And as for this, the amino acids, I should say, in these tails, such as that lysine and the arginine that was stated at the beginning of my presentation here, will be subject to the modifications that change how tight the chromatin will be packed, which I will talk about in a bit. So, to make sense of this information, here is a figure, and so this is the histone octamer, and as you can see, there are 2 of each of those histone molecules. There's 2 H2A, there's the 2 H2B, the 2H3 and the 2H4 that make up the octamer. So again, there's going to be a DNA double helix that will wrap around this octamer and come behind this H1 molecule that is in green here, where it will wrap around another histone until it becomes that 30 nanometer chromatin fiber all the way to eventually a chromosome. However, that green H1 molecule, that is going to be the one that facilitates that formation. From these histones, we are going to be able to see those tails that protrude from them, and these will be able to get tagged by the addition of the methyl and the acetyl groups, which I will talk about in the next slide. Lastly, this figure shows that this region here is named the linker DNA, and as you can guess, this linker DNA is going to be that region between the DNA double helix that's wrapped around the octamer. Considering this, the model here is also known as the beads on a string. The bead being known as a nucleosome particle that includes the DNA double helix that is wrapped around the histone octamer. And for the string connecting the beads would be known as the linker DNA, if I had to guess. So for those tails that are coming out of the histones, I've stated earlier that these will have amino acids in them, such as the lysine and arginine, that can be modified to where they will be where they can change in how the chromatin will be packed. And for those changes to happen, it will need to be either tagged by something like a methanol group or a acetyl group, not methanol, methyl group or acetyl group in a process called histone methylation or in acetylation. Concerning this, there is an enzyme called histone methyltransferase that will assist in the process of histone methylation, which is the methylation of lysine. In other words, this process will serve as like a signal for the activation or repression of a gene expression, depending on the lysine and histone involved. So, for example, I've read in the book that the methylation at lysine 4 in the histone H3 is where most active genes will be activated, whereas like lysine 9 in 27 will be for like gene silencing. Now, like, I don't really know the full details of these processes, but I do know that the methylation of histones will usually result in tighter packing. So, continuing on, there's going to be two more enzymes called histone acetyltransferase, which will assist in the acetylation process, meaning adding acetyl groups to the histone molecules to promote looser packing. And for the second enzyme, it's going to be called histone deacetylase, which will do the opposite function. With regards to these, with regards to the process of the acetylation of histone molecules using the enzyme histone acetyltransferase, this will promote the decondensation and the opening of the chromatin structure, so that it can allow like genes to be more accessible to be expressed, or even to be poised for expression. On the other hand, histone deacetylase will promote the closing chromatin structure, which could result in like the silencing of a gene. So, to do a recap of what a histone is, histones are going to be that spool for that DNA double helix to wrap around the beads on that string, so to say, so that they can be condensed and packed and to gradually make that desired chromosome structure. And that structure of that nucleosome bead will consist of a histone octamer, which allows for that DNA to be compactly wrapped. Additionally, the histone H1 molecule will bind to the linker DNA and promote the formation of a 30 nanometer chromatin fiber, where it will gradually turn to loops, eventually to heterochromatin, and finally a chromosome. Lastly, the tails will allow for the opening or closing of the chromatin structure, which can allow for a gene to be actively expressed or for it to be silenced. So once again, I just want to thank you for watching, and hopefully I at least explained the general idea of what a histone is. And yeah, I'll see you next time. Thank you.